Hi, my name is Leah Moylan, and I am from the Miller Lab at the University of Kentucky College of Medicine. I have the pleasure of sharing with you our project on assessing polygenic risk score accuracy for dementia risk stratification in the Alzheimer's Disease Neuroimaging Initiative dataset. So to go into a little bit of background, polygenic risk scores, or PRS, are quantitative measures of genetic predisposition. They tell us how likely an individual is to develop a certain trait or disease, or how protected they are against a certain trait or disease. Specifically, we focus on PRS for Alzheimer's disease, which can help direct clinical management of Alzheimer's disease. PRS are calculated using genome-wide association studies, or GWAS, which are large genetic studies that find correlations between single nucleotide changes in DNA and disease. The issue is that there are many GWAS available for AD, or Alzheimer's disease, and they all report different single nucleotide changes as correlated to AD. But you only choose one GWAS when calculating a PRS. This means that depending on the GWAS chosen, vastly different PRS results are obtained. The exact effects of GWAS choice on PRS are largely unknown at the moment. What we sought to find out is how GWAS inclusion impacts PRS accuracy and how combining PRS from multiple GWAS impacts overall risk prediction for AD. So the big key takeaway that we found throughout our work is that averaging polygenic risk scores across multiple studies may improve generalizability while maintaining high predictive accuracy. Now, we determined this using data from the Alzheimer's Disease Neuroimaging Initiative Dataset, or ADNI for short, which is a great resource for AD that has whole genome sequences, biomarkers, and other data on many individuals with Alzheimer's. We calculated polygenic risk scores for 808 individuals in ADNI using 17 different GWAS for AD. We calculated them using the PRS-KB, or Polygenic Risk Score Knowledge Base, which is a tool that our lab developed that allows for easy calculation and comparison of polygenic risk scores. We also calculated a PRS for each individual that was an average of all of their PRS produced from the 17 GWAS. This average was taken using the arithmetic mean. After cleaning our data, we analyzed it with various statistical tests. The first test of those being a Mann-Whitney U-test, which was used to determine if each GWAS could significantly differentiate between individuals with and without AD based on their polygenic risk score. So across the x-axis, we have each of the GWAS, and then on the far right is the arithmetic mean. Along the y is the p-value, and the red dashed line is the significance threshold for GWAS being able to correctly stratify AD risk. So two important things to note from this graph are that one, GWAS choice does greatly impact PRS accuracy. As you can see, with the great variation in bar height, depending on the GWAS chosen, a PRS may say an individual is incredibly at risk, or they may say that that same individual is incredibly predicted against AD. This highlights the importance of this work to determine new methods for GWAS selection that improve risk prediction accuracy. The second thing to note is that the dark blue bar on the right, the arithmetic mean bar, which represents PRS averaged across all GWAS, the important thing to note there is that it was able to significantly differentiate between disease status, and it performed similarly to the single most accurate GWAS. We also performed a chi-squared test focused on individuals within the top quintile of genetic risk. We again found a large variation in results based on GWAS chosen, and again we found that the average PRS across studies, again the dark blue bar on the right, was able to accurately predict disease status, but this time in individuals who are most genetically predisposed to AD. This indicates that averaging PRS across studies may be a more reliable method in predicting genetic risk for AD. In conclusion, our study found that there is in fact great variability in PRS accuracy based on GWAS choice. This variability may lead to overfitting, underreporting, or overreporting of disease risk. Additionally, we found that the average PRS across studies maintains high predictive accuracy for Alzheimer's disease risk, while also appearing to limit the potential biases introduced by selecting any single one GWAS. By integrating data from various studies, we reduce the influence of study-specific deviations overall enhancing the robustness of our predictions. Furthermore, our results emphasize the necessity for standardization in PRS computation methodologies, because currently there aren't really standardized methods for calculation. It is essential that these PRS calculations are consistent and validated for accuracy within the target population that they are trying to study. This standardization is paramount before PRS can be reliably implemented on a broader scale, especially in clinical practices, which is the end goal here. In the future, we plan to refine the inclusion criteria for which GWAS are considered in PRS calculations in order to further optimize the accuracy of risk predictions. We also plan to employ various machine learning algorithms to look into whether certain combinations of GWAS hold more predictive power than others. Maybe combining 5 of them instead of all 17 would be better, or something like that. By carefully selecting and combining GWAS based on rigorously defined standards, we can improve the reliability of PRS and therefore support their application in personalized medicine, ultimately contributing to a more effective early screening and intervention strategies for dementia. Our lab would like to acknowledge the International Society for Computational Biology and the Great Lakes Bioinformatics Conference. Thank you for listening.